Hello friends, I personally feel that biology students face difficulties while they study different biophysical techniques like NMR spectroscopy, mass spectroscopy, circular dichroism and all. So I have decided to create the videos or to create a series of videos on these techniques also. So in this video, we are going to discuss about circular dichroism. It's a small introduction for biology students. I will be discussing little bit maths of it, basic principle of it, and some very interesting examples of circular dichroism which we use in biological analysis. So we start from a very simple thing, that is Beer's Lambert law. Now we are having a test tube where our sample is filled and we are putting a light of intensity I0. Then this sample is absorbing this light and remaining light is passing through it, that is having the intensity of I. So according to Beer-Lambert law, log of I0 upon I is equal to A is equal to epsilon C into L. In this equation, I0 is the intensity of incident light, I is intensity of emitted light, and log I0 upon I is equal to A, that is defined as absorbance. So that is the main parameter of the whole system by which we are deciding how much light is absorbed by the sample. Now the absorbance is equal to epsilon Cl, where epsilon is molar extinction coefficient, C is the concentration of the sample in molarity, and L is path length. So this is the length by which the light is passing, that is equal to L, which is measured in decimeter or centimeter or different units, and the concentration is generally measured in molarity. So the Beer-Lambert law is giving us the information about absorbance or the light amount of light or intensity of light absorbed by the particular sample. So this equation is very important, guys, because we are going to use this equation in different scenarios. That is, A is equal to epsilon Cl, A is equal to epsilon C into L, where epsilon is molar extinction coefficient, C is concentration in molarity, and L is the path length. Now we see how many types of light we can use for this kind of experiment. So first we use unpolarized light. What do you mean by unpolarized light? Unpolarized light means where the waves or the light waves are having random vibration in all the directions. It is generally used for electronic spectroscopy. Second, the linearly or plane polarized light, where the light are doing vibration only in one plane. And third, circularly polarized light, where the polarized light is making circular vibrations or circular path movements, and the circularly polarized light is having two components. One, left-handed circularly polarized light, where the direction is anti-clockwise, and right-handed circularly polarized light, where the direction is clockwise. So these three kind of light we can use for spectroscopical purposes: unpolarized light in electronic spectra, linear or plane polarized light in ORD, or you can say optical rotatory dispersion, and circularly polarized light in CD spectroscopy or circular dichroism. So we will be focusing on this kind of light, where the circularly polarized light is having two components: left-handed component, which is anti-clockwise direction, and right-handed component, which is clockwise direction. Now we will be using this circularly polarized light to understand the basic principle of circular dichroism or CD. So we do the experiment again. So we are having now circularly polarized light, which we are putting on a sample, and the sample should be optically active, means it should be asymmetrical, or you can say it should be having a chiral center like alpha helix or amino acids or carbohydrate. So an optically active molecule, when it will interact with circularly polarized light, then because of its symmetry, This molecule or these molecules absorb left-handed and right-handed light differentially. It means absorption of left-handed and right-handed light by an optically active compound is not identical. Sometimes some compound absorb more the left-handed light and less right-handed light, or vice versa. Opposite condition will be there where some compounds will be absorbing right-handed light more than left-handed light. So what we are getting from this picture is, when we are having a sample which is optically active, then it will be absorbing the left-handed light as well as right-handed light, but the absorption for both of these two light is not identical. Left-handed can be more than right-handed, or right-handed can be more than left-handed. This concept or this situation is called as circular dichroism. So if somebody asks you what is circular dichroism, then in one word we can say that when we put a circularly polarized light on an optically active molecule, then this optically active molecule absorbs left-handed and right-handed light differentially, and this difference in absorbance is called as circular dichroism or CD. So CD is nothing but what is the difference between the absorption of left-handed light as well as right-handed light. So when we want to measure the circular dichroism, we are measuring nothing but difference in the absorbance A, which we have seen in the Beer-Lambert law, that we are measuring the difference in absorbance, and this difference is called as circular dichroism or CD. Now we see how the units are taking part in the circular dichroism. We see some of the mathematical part of this CD spectroscopy. Now, how do we measure circular dichroism? So simple. A sample is having absorption on y-axis and wavelength on x-axis. So the blue is showing the absorbance for left-handed light, and the red is showing the absorbance of right-handed light. So we can say that in this case the absorbance or absorption is different for these two lights. It means this sample is showing dichroism, and the final resultant light will be the subtraction of left-handed as well as right-handed, left-handed and right-handed light. So the circular dichroism CD can be measured by delta A, where delta A is equal to AL minus AR. AL is the absorbance of left-handed light and AR is the absorbance of right-handed light. So as I said in last slide, circular dichroism is nothing but difference of absorbance between left-handed light and right-handed light. So delta A is equal to AL minus AR. Now, according to Beer-Lambert law, we know that the equation for A is equal to epsilon multiplied with C and multiplied with L, where epsilon is molar extinction coefficient, C is concentration in molarity, and L is the path length. So I can write the equation. Of A is equal to epsilon CL. So the delta A will be epsilon L for left-handed light, C into L. Since the test tube is same, sample is same, so the concentration and length will be same. So epsilon L C into L minus epsilon R C into L. So we just have kept the value of AL, that is epsilon L into CL, and AR epsilon R into CL. It is very simple. Now in next step, the CL part will be common, and I can write EL minus ER, or epsilon L minus epsilon R, is equal to delta epsilon. So delta A is equal to delta epsilon C into L, where C is concentration and L is the path length. Now if the concentration is 1 molar and path length is also unity, means 1, then delta A will become equal to delta epsilon, that is circular dichroism. So the circular dichroism can be measured as the difference between absorbance of left and right-handed light, as well as the difference between the molar extinction coefficient for left-handed as well as right-handed light. So it is very simple. If absorbance for left-handed light is more than right-handed light, then the circular dichroism will be positive, and if absorbance of left-handed light is less than the right-handed light, then circular dichroism will be negative. It's very simple to measure. So circular dichroism is measured in terms of difference in absorbance as well as difference in molar extinction coefficient of left-handed light and right-handed light. So till now we have understood the basic concept of circular dichroism as well as how we are going to measure it. There is one more unit of measuring the circular dichroism, that is ellipticity, and that is one of the typical part or little bit difficult part to understand, but I will try to explain it in easy words, or you can in an easier manner. So we can see here that again we are doing the experiment. We are putting circularly polarized light with two component, left-handed component as well as right-handed component on the sample which is optically active. Now we are getting this kind of situation. So what is this diagram? So the blue line is the amplitude of right-handed light which is coming out of the sample, and a red circle is showing the amplitude of left-handed light. So by looking at these two circles, I can say that the sample is absorbing more left-handed light than right-handed light. That is why initially the amplitude was same for both, but after absorbance the amplitude of left-handed light is decreasing and amplitude of right-handed light is more than left-handed light. Why? Because left-handed light is absorbed more than right-handed. So since the absorbance of left-handed light is more, that is why after absorption the amplitude is decreasing, and in this case we are having two different amplitude, less amplitude for left-handed circularly polarized light and more amplitude for right-handed circularly polarized light. So when we are having this condition where two lights which are having different amplitude are coinciding with each other, then we get the formation of an ellipse, and the angle which is created by this ellipse is called as theta, or you can say ellipticity. So theta can be measured as circular dichroism. How? If there is more difference in the absorption of left-handed light and left-handed light and right-handed light, then there will be more value of theta, or you can say ellipticity will be more. And if the absorbance is, or the difference of absorbance is less between these two components, then there will be less ellipticity, or you can say there will be smaller value of theta. So I can say more theta means more difference of absorbance or more circular dichroism, or less value of theta means less ellipticity, or you can say less difference in absorbance, or finally you can say less circular dichroism. So this theta can be converted into delta A also. So all the units of circular dichroism can be interconverted. So the equations are delta A, that is circular dichroism, is equal to theta upon 32.982. So if we know the ellipticity, we can find out the difference in absorbance, or we can find out circular dichroism in terms of absorbance. Now there is molar ellipticity, one more component or one more unit, that is nothing but 100 into theta divided by concentration and path length. So if you are mentioning, or if you are showing theta in terms of concentration and path length, then it becomes molar ellipticity, and there is relationship between molar ellipticity, that is square bracket theta, and delta epsilon, that is again circular dichroism, but in terms of molar extinction coefficient, that is delta epsilon is equal to molar ellipticity theta divided by 3298.2. So guys, these equations are very important because by this equation the ellipticity can be converted in the difference of absorbance, or by this equation molar ellipticity can be converted into delta epsilon, that is again circular dichroism. So we can measure the circular dichroism in 3 units or in 3 methods. First is delta A, that is difference of absorbance. Second is delta epsilon, that is difference of molar extinction coefficient. And third is theta, that is ellipticity. If delta A, theta or delta epsilon values are more, it means more circular dichroism, or you can say there is more difference in absorption. If values of delta A, theta and delta epsilon are less, then you can say there is less circular dichroism, less difference in the absorption of left-handed and right-handed light. So till now we have understood the basic principle, basic idea, different units, basic maths of CD. Now we see different examples. So here I am keeping it very simple. I will be explaining you mainly the circular dichroism of proteins for all the biology student, and I think you will be enjoying the things. So this is a typical graph. What is happening here? So it is looking a little bit complex, but it is very simple to understand. On x-axis we are having wavelength in nanometer, and on y-axis we are having ellipticity, that is theta. So what I am finding here, this is the line which is a dark black line, that is for alpha helix. So alpha helix is having two circular dichroism band. One is positive, that is here, second is negative, that is here, and third is negative, that is here. So you can say there are 3 bands of circular dichroism for alpha helix, 1 positive and 2 negative. Positive means the absorbance of left is more than absorbance of right, and negative means absorbance of left is less than the absorbance of right. So I can say that for alpha helix we are getting 3 bands of CD, 1 positive and 2 negative. Now this dotted blue line is for beta sheet. I can say that there is 1 positive and 1 negative band. And finally we are having a random coil which is not alpha helix as well as which is not beta sheet. We are having a strong band at negative and very less ellipticity which is positive. So these 3 lines can be used as standard curve of CD spectroscopy for alpha helix, beta sheet and random coil. Now again we repeat it. Alpha helix has a negative bands at 222 nanometer and 208 nanometer, which are here, 208 and 222, and 1 positive at 190 nanometer, that is here, 190. For beta sheet we are having negative band at 218, that is here, and positive one at 196, that is here. For random coil we are having a positive band at 212, that is here, and negative one we are having at 195, that is here. So you just remember this graph, that for alpha helix we are having total 3 bands, 1 at positive and 2 are negative. The values are 190, 208 and 222. For beta sheet we are having 2 bands, 1 at 196 and 1 at 218. And for random coil again we are having 2 bands, one is negative, one is positive, positive at 212 and negative at 195 nanometer. So this graph we are going to use again and again, and these values are very important. They can be asked in your exams, or you can use these values to explain the CD or circular dichroism. Now we are having this graph, standard curve. We are going to use this as a standard curve again and again, and we are going to understand this very difficult thing which is looking like here. So we are having a blue line, that is chymotrypsin, and chymotrypsin is an enzyme which is having nearly all the beta sheets. So this is the structure here, but when we look at the CD, so this is the CD here for chymotrypsin, and it is actually matching with what? Beta sheet. Then we are having lysozyme, which is containing mixed alpha and beta sheets. Lysozyme is having alpha helix as well as beta sheet. So this is the red line. So it is having alpha helix nature also, and it is having beta sheet nature also. Then there is another enzyme, triose phosphate isomerism, which is generally or mostly alpha, and there are some beta also. So it is showing the mixed CD spectroscopy. And finally myoglobin, which is all alpha. So you can see here is the myoglobin line, that is positive band. It is similar to positive band of alpha helix. Then two negative bands which are similar to alpha helix here. So in CD spectroscopy we are having nothing. We are using this graph as standard curve. Then we are taking unknown sample and we're doing CD spectroscopy, and then we are matching the bands with previously present standard curves. So it is like curve fitting method. So for unknown protein or for a new protein, you just take out the band values for CD and then you try to fit it with alpha helix, beta sheet and random coil proteins or standard curves. Then you see that what is the percentage of alpha helix in a given protein, what is the percentage of beta sheet in this given protein. So this example is nicely explaining it, where 4 types of enzymes are taken, all beta, mixed alpha and beta, mostly alpha and all alpha, that is myoglobin. So we can see here we are getting very good similarity between these graphs and these standard curves, and this method can be used for any CD spectroscopy. There are software, there are algorithms. So when you run a unknown sample for CD spectroscopy, then those algorithms or those programs, they just fit the values of bands for unknown protein with standard curve, and they tell you what is the percentage of alpha, what is the percentage of beta sheet, what is the percentage, whether they are mixed with alpha or beta, or whether they are alternatively alpha or beta. So we can get very good information, very good information about the structure or the percentage of secondary structure, percentage of different secondary structures in the protein. So it was a very nice example to discuss and very nice example to understand. So in next example, we are going to use the same standard curve. Now we are going to compare it with another interesting example. So we have taken a same protein with 3 different temperature conditions, 0 degree, 50 degree and 75 degree. So at 0 degree we are getting this blue line. So this blue line you can see exactly matching with the alpha helix, and I can say that at 0 degree this protein is having 100% alpha character, or you can say nearly 100% alpha character. Now when we increase the temperature to 50 degree, then this alpha character is decreasing because this positive band is decreasing. We are not getting positive ellipticity at the same wavelength. So we are having reduction in alpha character. You can say the helix is opening on higher degree of temperature, or you can say protein is getting denatured or protein is getting unfolded, and at a very high temperature of 75 degree, then whole alpha CD spectrogram or whole alpha CD spectra is gone, and we are getting very less ellipticity. So as I said, if you are having helical structure, you have higher value of ellipticity, it means there is more circular dichroism, and when you are having a linear structure, there is very less value of circular dichroism. Right, so this can be understood by this very interesting example. Now we take one more very interesting example, which is my personal favorite. I found it in a very interesting research paper, and I am amazed by this example, that how the CD is helping in understanding of the molecules and all. So in this example also we are going to discuss the same standard curve for alpha helix, beta sheet and random coil. And they have designed a molecule which can absorb the light and can change its conformation. So this is the conformation of molecule in dark, and this is the conformation of molecule in light, right? So they have attached this molecule to a protein. So here is this molecule attached to this protein, and this is the condition of dark, right? So in dark, this molecule is a trans molecule. So when it is bound with the protein in trans condition, the protein is less helical, you can say. And once you give it light, the molecule is converting into cis conformation or cis condition. Then once the molecule is converting into cis configuration, then the protein is becoming more helical. So in dark, because of this molecule, protein is less helical, and in light the protein is more helical. And when we check the CD spectroscopy of this protein bound with this molecule in dark as well as in light condition, we get this kind of graph. It is very interesting. So in dark, the protein is less helical, so it is not showing the alpha helix CD spectra. So it is more like a random coil here, you can see, right? And once you are giving the light exposure, the molecule is changing trans to cis, and with this change in trans to cis, the protein is getting more helical, protein is becoming alpha helix, and we are getting now a very good alpha helix CD spectra. So in dark it is not alpha helix, and in light it is alpha helix. So it is beautiful example to understand that how you can maintain the structure of helix, or how you can change the helical structures based on light. So you are using a molecule which is responding to light, and this molecule is changing the helical nature of the protein, and which you are measuring with CD. This example is really one of my favorite, and I hope you have understood this example. Okay, now other than protein, we can also measure the circular dichroism for DNA molecule. So this is the example. Again, on x-axis we are having wavelength, and on y-axis we are having circular dichroism. So this is for a double helical DNA, and when you heat it up it gets denatured. So when you are heating up, then helix nature is lost, and since the helix nature is lost, then circular dichroism is decreasing. So this is the line, dark line at normal temperature, and these dotted lines are on different higher temperature where the helical DNA is opening, or DNA is losing its helix property. So as I said, the circular dichroism is dependent upon the helical nature of the molecule. More helical nature means more value of circular dichroism, and here it is self-evident that at normal temperature DNA is helix, so we are getting good circular dichroism, and at higher temperature DNA is denaturing, or you can say the helix of DNA is opening, so we are having less value of circular dichroism. It is again very interesting example where we are understanding the circular dichroism of DNA. One more interesting example I found, that there is a dye which is called as acridine orange, and it can bind to DNA. So we have measured, or people have measured the CD of this dye acridine orange. So once it is bound to DNA, then it is showing helix nature, or it is showing circular dichroism, you can say. So at 0.0 means it is not bound with DNA, so there is not a good value of ellipticity. 0.03 means binding of the acridine orange with the DNA is increasing, so we can say the ellipticity is increasing, or the absorbance is increasing, or you can say the circular dichroism is increasing. More concentration of acridine orange bound with DNA means more circular dichroism, and very high circular dichroism if we are having very high binding of acridine orange. So based on binding of acridine orange, we can say that okay, the circular dichroism is increasing. So these things can be used for circular dichroism for different application, where DNA is binding with proteins and protein-protein interactions, and drug DNA interaction or drug protein interaction, secondary structure determination. So circular dichroism is one of the very important biophysical technique which is giving us a very good data about the structural aspects of biomolecules in very less time, and you can say with less mathematics than other spectroscopic techniques, right? So I hope you have understood the basic principle of circular dichroism, mathematics of circular dichroism, units of circular dichroism, and different examples. And I hope you are understanding what I am trying to say here, and in next videos of the same series I will be making videos on NMR or mass spectroscopy or electronic spectra or fluorescence, different biophysical techniques. So thank you guys, thank you very much for your time. For this whole presentation or whole video, I have used so many different things which are very interesting to read. There is a very good paper by Greenfield that is using circular dichroism spectra to estimate protein secondary structure. Then there is another paper where circular dichroism signals for GCN4P1 protein. Then there is another paper with Smart and Woolley that is published in PNAS in 2000, and a very good book that is Spectroscopy for the Biological Sciences by Gordon G. Hams. And this book has given me too many insights of circular dichroism, and that is why I decided to create a video where I can explain the circular dichroism to the biology student, that how they can use it